Bismillahirrahmanirrahim. Welcome back friends, welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to see another biochemical test, that is about Casine Hydrolysis Test. Now what is Casine Hydrolysis Test? First of all, let me explain the casein. Casein is actually the milk protein. We also call it phosphoprotein. It is actually the macromolecules that is mostly found in protein, that is mostly found into the milk, and it is available 85% into the milk. Okay, now this type of the test is specially used to isolate the bacteria from the dairy products. Okay, now during this test, what's going on, the casein, as we better know that it is a macromolecule and it cannot be fit into the cell of the bacteria. So what's going on, the bacteria will release an exoenzyme, that is the peptidase, protease or casinase. So when they release these exoenzymes, so it will hydrolyze or degrade the casein into amino acid. Then the bacteria will easily uptake it and use it. This is how the Casine hydrolysis test is working. Then we have the objective. So this test is specially used to determine the ability of the bacteria that degrade the casein by the action of casinase exoenzyme. The requirement, so in this test we required skim milk, agar media, personal protective equipment, spirit lamp and petri dishes. We need a fresh bacterial culture, autoclave and incubator. The composition of the media is skim milk agar, we need 28 gram, yeast extract we need 2.5 gram, tryptone we need 5 gram, glucose we need 1 gram, agar we need 15 gram. So we will mix all these ingredients in 1 liter and then we will autoclave it. The principle says that what will going on, that we better know that casein is the protein and it is white in color and is insoluble protein, and due to which the media are opaque and milky white in color. Okay, now as we already talked, that casein is the macromolecule and the bacteria cannot utilize the casein molecule directly for the metabolism. So what's going on, the bacteria will produce enzyme that will degrade or hydrolyze the casein into smaller amino acid and peptides, then the bacteria will take it. So when the bacteria hydrolyze it, so what will happen on to the media, the proteolysis, when we want to see the proteolysis, so onto the media we get as a clear zone, clear transparent zone, and this clear transparent zone is actually the positive result, and it indicates that the casein is hydrolyzed and the bacteria utilize the casein. Okay, now the tryptone yeast extract we are using in this media, it is specially used to provide nutrition for the bacterial development. We use agar is specially used for the solidifying agent, and we use distilled water that will mix all the ingredients. So this is the whole process and this is the principle of the Casine hydrolysis test. Then we have the result. So first of all, let me explain the procedure. Now the procedure says that first of all you need to prepare the media. Once the media is prepared, then you need to sterilize the media. Now the media is sterilized, then you will pour the media into the petri dishes. Once the media is solidified, okay, because it is agar, so once it's solidified, so then you will take the heavy inoculum from the fresh bacterial from the fresh culture and will streak it onto the media. Then after that you will incubate the plate for 24 hours, and after that you will check the development of the zone onto the plate. This zone development actually indicating that the casein is hydrolyzed, and the zone around the bacteria is actually indicated that the casein is hydrolyzed and the bacteria utilizes. Then we have uses. So this test is specially used to find out the casinase activity. Okay, and it is also used to find out the casinase activity through which the bacteria can grow into the milk. This test is also used to differentiate Bacillacea, Enterobacteria and other family. We can also use this type of media to isolate aerobic actinomycete. So this is the uses of the Casine hydrolysis test. Then we have the result. Now this is the result. As you can see, this first picture is the positive result, this is the negative result, and this is the simple control. Okay, now this is the plate. As you can see, the plate will be look like in this one, opaque, white, creamy like color, just like milk. When you streak the bacteria and when you get the zone, it means that casein is hydrolyzed. Okay, so this is the positive. This may be the bacillus, this may be the pseudomonas, this may be the lactobacilli, serratia. Okay, so those bacteria that are the positive, we have bacillus subtilis, bacillus cereus, pseudomonas, lactobacillus, and we have actinomycetes, etc. Now these are the negative result, and in this case we have different type of the bacteria like E. coli, enterococcus faecials, etc. And this is the simple plate, so it will be look like in this one. Then we have the limitations. So while determining the bacteria or bacteria species, you need to perform other biochemical tests that is really necessary. Okay, so it is not important that we will depend on one test. Next, we required longer incubation in this test, and this test the fastidious organism cannot grow, so this in the medium, and it is very difficult to find out. So this is all about the Casine hydrolysis test. I hope this video is helpful for you. Thank you so much for watching.